In this second video over our material over chapter 15, we're going to take one more big time concept, the idea of half-life, and then we're also going to add to it just a couple of applications about nuclear chemistry that are going to be relevant and important to us, especially in our career field. So let's take a look at all right, so half-life. Half-life is just the amount of time that it takes for half of our radioactive material to decay or disintegrate away. Now, what's really interesting about radioactive decays is that the half-life for those decays is a set number, meaning it takes the same amount of time for half of the material to go away if I have 1 kilogram of it, or half of a kilogram of it, or a quarter kilogram of it, or any amount of it. In this case, 1,620 years is the half-life. So half of it will go away every 1,620 years. Now we can calculate the fraction that is left over after a series of half-lives by taking the number 1/2 and multiplying it by itself, however many half-lives have elapsed. So after 1 half-life, 1/2 to the first, I would have half of the material left over. After 2 half-lives, 1/2 squared, I'd have 1/4 of the material left over. After 5 half-lives, 1/2 to the fifth, I'd have 1/32 of the material left over. So let's look at this question. I have gold 128. It undergoes a beta decay to give mercury 128. The half-life is 2.7 days. What percentage is left after 8.1 days? Well, to answer this question, I need to figure out how many half-lives have gone by. And to do that, N is equal to the time divided by the half-life time. So 8.1 days have elapsed. 2.7 days is the half-life. 8.1 divided by 2.7 is 3. I have 3 half-lives that have gone away. And so the way I do it is I start at 100%. 1 half-life turns that to 50%. A second half-life turns that to 25%. A third half-life turns that to 12.5%. Now, you could have also come up with that in the same kind of way by doing your 1/2 to the third, which would give you 1/8, and 1/8 is equal to 12.5%. You could have come across that either way. For me, I just kind of like the being able to kind of see where I started and how it goes to where I ended. And all I did in each step was take that number and divide it by 2. So 100 divided by 2 gives me 50. 50 divided by 2 gives me 12.5 or 25. 25 divided by 2 gives me 12.5. Now, one of the most common uses for this is something called archaeological dating, radiological dating, radiocarbon dating. And this is where we can use carbon-14 and its presence as a way of measurement. The concept here is rather simple. When things are alive, they are respirating, they are moving, they are eating, they are drinking, and they are constantly replenishing their cells with what they eat and what they drink. And so that includes carbon. So your body naturally has a certain percentage of carbon-14 in your body. Now, we know carbon-12 is by far way more abundant, and carbon-12 is very, very stable. But carbon-14 is not. And so the decays that happen inside of your body of carbon-14, they're relatively unnoticeable. And the reason why is because even if it does decay, you eat something and you naturally get a little bit of carbon-14 back in your system from what you eat. Because everything has trace amounts of carbon-14 in it. But When you die and you no longer are metabolizing, you're no longer replacing the carbon-14 that is decaying, the carbon-14 concentration in your body starts to diminish, starts to deplete. And because nothing's in there to replace it, to push it back up, it naturally is going to decline and decrease over time. Now, this particular process has a half-life of about 5,730 years. So what we can do is we can compare baseline carbon-12 versus the amount of carbon-14 that's in the system. We can compare them to each other, and we know what that ratio is for living things. For non-living things, we can compare, okay, this is where it was, this is where it is. How long did it take to go from here to here in concentration? And based upon that, we can use that to measure half-life. Well, we know the half-life. We can use that to measure how much time has elapsed based upon the ratio of one to the other. So a leather strap is found in an archaeological dig. It's found to have 25% of carbon-14 compared to what is in normal living tissue. If the half-life is 5,730 years, how old is the leather? Well, this one we're kind of working backwards on compared to the previous problem. We're at 25%. One half-life would have got us to 50%. A second half-life would have got us all the way to 100%. So we know that 2 half-lives have elapsed. Each half-life is 5,730 years. So 2 times 5,730, 11,460 years old for that leather strap. So this is obviously a simplistic example. Very rarely do we find that the numbers are so clean and easy for things in archaeology, but we know that there are ways to do a more sophisticated comparison that give us a little bit more accurate of a number if the numbers aren't so clean. And that's what's used in a lot of kind of commercial and forensic laboratories for this kind of idea. Now, what are some other applications? So radiocarbon dating is a really cool one. What are some other ways that we can use radioactivity and isotopes to do cool things? Well, one of the things that they can do is medical applications. For example, power generators. The isotope plutonium-238 is used to power pacemakers. And so it delivers an electronic pulse as it decays. That is what allows a pacemaker to do its thing. We can use it for medical diagnosis. So by having patients ingest small amounts of radioactive nuclides, we can track the movement of those nuclides into different parts of the body, use that to determine how body systems are functioning, whether or not they're functioning properly. For example, technetium-99, which is a metastable nucleus, we can find through gamma radiation. And doctors use that gamma radiation that it emits to help locate tumors inside of the body. So how do we do something like that? Well, looking at this picture here, notice this white space here in the dot diagram. Iodine-131 gives off signatures that are being able to pick up by instruments. The iodine-131 was being absorbed by this tumor, and so it was not emitting the proper tracer. And they were able to determine that it was present here based on the fact that the iodine was not getting picked up the way that it should have. Other medical applications, we talked about positrons, positron emission topography, PET imaging, PET scans. Patient takes a small amount of a positron emitter. The positron emitter goes through different parts of the body, goes into the living tissues, and we can see, based on signatures, a PET scan can detect abnormalities in the tissue without actually disrupting the tissue itself. So PET imaging is very useful in that regard. We've seen radiation used in cancer therapies. This has been done for years. Radiotherapy, radioactive nuclides, radiation therapy. We give patients radioactive materials in high doses to treat cancer. The cancer cells absorb the nutrients that contain these gamma emitters. The gamma emitters become concentrated in the cells, and the gamma radiation destroys the cancerous cells. And it does so in a much higher rate than normal cells are. Now, granted, there's usually collateral damage that comes along with radiation treatment therapy, where the cancer cells are destroyed in some cases, but so are some of the healthy cells around them, but at a much lesser degree, to a much lesser extent. Some specific examples of this: iodine 131 is used for thyroid damage. Gold 198 rather is used in the treatment of lung cancer. Phosphorus 32 has been used for eye tumors. There are other examples out there as well. Some other applications. Irradiation. This is a technique that has picked up some steam. It's still very, very not well defined. There's still a lot of kind of anxiousness about using gamma radiation. But one thing that it has been done has been that we put gamma rays and expose them to certain foods. And what happens is the food isn't harmed by the gamma radiation, but the microbes that are on the food are. And so the mold, the bacteria, whatever is on that strawberry at that time gets killed by the gamma rays. The strawberries themselves are unharmed. And we can see the difference between them in picture B. Now, if this is the kind of thing that scares you, do be advised that all things that are treated with radiation have to be marked. This is called the Radura symbol, right here. This is called the Radura symbol. Anything that is treated with radiation has to be marked with the Radura symbol. To not mark it would be illegal and would be very, very, very costly to the company if it was ever discovered. So that is a safeguard that is put in place by the FDA, by the USDA, to let people know what they are buying. So if you see that symbol, it means that the food has been treated with radiation. If you do not see the Radura symbol, then it means that the food was not treated with radiation. Very easy way to tell the difference. The last thing we're going to talk about, kind of a big picture topic here, is the idea of nuclear energy. So one of the byproducts of the nuclear age and the age of nuclear weapons has been that once we developed the atomic bomb, and we saw all the different things that nuclear reactions can do from a destructive standpoint and how much energy that is released, it became a symbol to try to figure out, well, are there ways that we can harness that energy in a good way? And so the initial atomic bomb was a fission bomb. And in fission, what we are talking about is the splitting of a heavy nucleus into two or more light nuclei. How this happens usually is you take a substance like uranium-235, you hit it with neutrons. The neutrons get absorbed by that nucleus, form uranium-236, which is highly, highly, highly unstable. And that highly, highly unstable nucleus breaks apart, and we end up getting krypton and barium. And what's really, really interesting is we get more neutrons as a result. Now, why is this part interesting with the neutrons? Well, because these neutrons are ejected at high speeds, which means they can go out and they can interact with other uranium nuclei. And so the action of one neutron generates 3 more neutrons that can go out and cause the decay of 3 more uranium nuclei. And so this is kind of pictorially what I was just saying. the neutron comes in, we form an unstable nucleus. We get the unstable nucleus to split into 2 more stable nuclei. We also get 3 more neutrons that can go and replicate this process, and a whole lot of energy. If there is enough uranium present, then what we can get to happen is a chain reaction. And a chain reaction would be when those neutrons that are ejected by this reaction go out and form other reactions as a result. So in order for this to happen, you have to have enough fissile material present to get that reaction to occur, to get that chain reaction to occur. And that minimum amount of fissile material is called a critical mass. And so if you hear about critical masses from a standpoint of nuclear reactions, that's what we're talking about. Most nuclear power plants operate at lower masses of exposed nuclei than the critical mass as a safety precaution. When we see critical masses, we usually think about them in terms of bombs, because bombs will have critical masses or greater than critical masses of the fissile material in order to get the reaction to run out of control very quickly and produce all of that energy, all of that radiation, cause that mass destruction. But since most of our work with fission now involves nuclear reactors and not nuclear bombs, let's talk a little bit about how that works. So a nuclear power plant uses fission in this way to create energy. So we have uranium rods inside of a reactor. The uranium rods are exposed to neutrons. They undergo their fission reaction in a relatively controlled manner. And as a result, the energy that they create is harnessed and converted into electricity. Now, one of the safeguards that is in place are these boron-10 control rods. Boron-10 is a neutron poor nucleus of boron. It actually prefers to be boron-11. And so when the reaction starts to run out of control a little bit, boron control rods get inserted into the reactor to absorb the extra neutrons and slow the reaction down. But the whole idea of a nuclear reactor is basically it's a big water tank. The nuclear reaction occurs, the energy is outputted, and it's really just outputted to turn water into steam. The steam then turns a turbine, which turns a generator, which makes electricity. The remainder then goes through a condensing cooling station. And so this big picture, the image that we usually associate with a nuclear power plant, that big cone-shaped tower, that's a cooling tower. And all that cooling tower emits is steam. So as the cooling loop is doing its thing, it is cooling down the water inside of the reactor so that the water can go back into the reactor, get converted back into steam, turn into more electricity. And the net result of that steam, of that reaction in the cooling tower, there's no reaction. It's just the hot water is running into particles of the air, condensing them. We're getting steam off of that as a result because the water going through this cooling loop is considerably warm. And that's it. That is all for this particular chapter. And so thank you for watching this video. Thank you for your attention this semester, and I will see you in class next time.